Okay, so this is going to be the second video for chapter 11. Um, so we talked a little bit about the dynamics of forming spheres and a bilayer. I want to talk a little bit about fluidity. And fluidity has a lot to do with the composition of the lipids that the phospholipid is made out of. If you recall, I said that the the saturated fatty acids are much more dense than the unsaturated fatty acids. And you probably already know this from your own experience that fats derived from animals are more saturated and are more dense than fats that are derived from plants. And so in order to have a consistent fluidity, one of the strategies that cells use is to use use steroids, cholesterol, to rivet together the lipid portions of the bilayer, and and that way they get a more consistent fluidity. So if uh, if there was no cholesterol, then our cell membranes would be about the fluidity of olive oil. And anybody who's owned olive oil knows that you put it in the refrigerator, it's solid. You keep it at room temperature, it's a liquid. We wouldn't really want our cell membranes to be either solid or liquid. We want it somewhere in between. And the cholesterols are going to do that for us. Um, so remember what saturation is do you have all of the possible hydrogen bond or hydrogens bound covalently to carbons that you can have. If if you are not fully reduced, then you're going to end up with some double bonds in between the carbons, and those double bonds create a kink. And so unsaturated fatty acids, like those that come from plants, have these kinks in them, and those that causes the lipid to take up more space, and so it's just not as dense. And so, like for instance, butter is solid at room temperature, um, depending on the temperature of the room, but it's solid at room temperature, and vegetable oil is liquid at room temperature. Um, and that has to do with the length of the carbon chain and the number of double bonds that are there. So, so in order to overcome this fluidity problem that we have, we use cholesterols to link the the fatty acid tails of the phospholipid to each other as kind of like joists in an attic. So so in addition to the phospholipids, the membrane also has proteins, and there are lots and lots of different kinds of proteins that are associated with the membrane. Um, they can have lots of various functions, like helping things to cross the barrier, like um, small molecules or ions. Um, they could be enzymes. They can be there's lots of different proteins that are associated with the membrane, and most of the ones that are integral membrane proteins are going to cross the bilayer using alpha helices. Alpha helix is the most common secondary structure for crossing the bilayer. You can do it with beta sheets, but that really restricts your size and therefore the types of proteins that you can make out of it, because you have to put like 11 of them into a barrel to create a beta barrel, and and so that's going to limit the versatility of the proteins that you can make out of bilayer bilayer crossing beta sheets. But bilayer crossing alpha helices, much easier. Um, so we we have a lot of trouble studying integral membrane proteins because of their lack of solubility. It's really hard to solubilize proteins that are that hydrophobic. We have to use detergents, and um, and so we're going to talk a little bit about amphipathic and what that means. So what are some of the things that these proteins can do? They can be transporters, moving ions in and out. They can be anchors for linking the cytoskeleton across the membrane to the extracellular matrix, creating um, cell sheets, uh, um, and that's going to be important for things like organs. They can be receptors for communication. A extracellular signaling molecule binds to it and causes some sort of signaling cascade inside of the cell so that the cell can react to its environment. Or they can be enzymes, all of the enzymes associated with oxidative phosphorylation and the generation of uh, energy are all membrane-bound proteins. So the diversity of function of integral membrane proteins is really vast. Um, they can be lots of different structures too. Like I said, the most common is an alpha helix, but you can also have beta sheets, and so that's beta barrels, and so that's how we're going to cross the membrane. But you don't have to be crossing the membrane to be associated with the membrane. You could be a protein that is associated with a protein that crosses the membrane. Those are called membrane associated proteins. So they're attached to the membrane via another protein. They can also be lipid linked. So all of these glycosylations, stimulations, myristylations, GPI anchors, all of these post-translational modifications can cause the protein to be associated with the membrane. And they don't have to completely cross the membrane. You could just be associated with one leaflet of the membrane. So like I said, the complete structure of these proteins is still relatively hard to tease out, and the reason for that is um, because of the difficulty of solubilizing these proteins. The way that we solubilize these proteins is by using detergents. Detergents like SDS or Triton X, in this case Triton X100, but there's lots of different densities of Triton. And what these detergents are is amphipathic molecules. They have a a um, a side that is charged and so polar and will interact with water, and a side that is uncharged and non-polar, and like a large carbohydrate chain is going to be kind of greasy, and it's going to associate with the hydrophobic regions of the protein. And so these hydrophobic side and a hydrophilic side allows for the detergent to completely decorate the protein. Um, they get all over the protein on their hydrophobic sides, attaching to the hydrophobic portion of the protein. And then their hydrophilic side um, is going to be able to interact with water and solubilize the protein. And so that's how we study these proteins, is by using detergents to pull these proteins out of the membrane and then into a test tube where we can maybe start to look at their structure, slowly pull the water away, and maybe make crystals of the proteins, and then use X-ray crystallography. Or we can study their function by putting them into artificial membrane situations. Remember, I said that it was pretty easy to make micelles, and so that's one way to study the proteins, to study their function. So how do we know that membrane proteins have, or the membrane has fluidity? Well, the way we determined that was by using dyes like rhodamine and fluorescein. And if you take one cell and you cover it in rhodamine and take another cell and cover it in fluorescein, um, then you fuse these two cells. Over time, it will go from a cell that has a red side and a green side to a cell that is all yellow, mixing the red and the green um, fluorescent dyes. And so that's how we can prove that that there's a free movement side to side of the entire membrane. Another way that we've done it is by taking green fluorescent protein um, and these green fluorescent proteins, you put them into the membrane um, and uh, and then you use photo bleaching. So you shine a laser on them and then they stop fluorescing. And then over time, the fluorescence will recover um, because the fluorescence of the GFPs on either side of the bleached spot move into that spot. And so this is called fluorescence recovery after photo-bleaching and or FRAP. And it is a method for studying how fluid a membrane is. So there are other ways that we can limit mobility of proteins within the membrane, and those have to do with anchoring. And so if we anchor the protein to the cytoskeleton, then it's not going to move side to side very easily. Or if we anchor it to the extracellular matrix, it's not going to move side to side very easily. Cell adhesion molecules that are interacting with cell adhesion molecules on another cell are not going to be able to move very easily. And then um, in cells that are asymmetric by nature, like say your gut epithelial cells, you have an apical side and a basolateral side. And so what we do there is we have a specialized cellular junction called an occludens junction. And the occludens junction doesn't allow for things to move between the cells, but what it also does is it sets up a barrier that it doesn't allow the apical proteins to go to the basolateral side. And this is very important for the epithelial cells of the gut because there are transporters that you only want um, transporting stuff from the lumen of the gut into the cell. You wouldn't want that same protein on the other side of the cell, on the basolateral side. Same thing with the basolateral proteins. You wouldn't want them on the apical side. So we use these anchors and the cortex in all of our cells. And for an example, red blood cells have a very stereotypic shape. And that shape is created by actin interacting with spectrin, vinculin, all of these junction proteins that create this beautiful hexagonal matrix on the inside of the cell. So these are the cortical proteins, and they create this gorgeous shape of the red blood cell. So some of these proteins, band 3.1, band 4.3, spectrin A and B are proteins from our first lab. So remember those. Okay, so that's all I've got for this video.